Well, we have left our discussion at the point where we are familiar with the optical microscope, the basic principle of the optical microscope, and what is the essential difference between the optical microscope and electron microscope. So now let's come to this point, what we are talking about here about the instrumental setup. I already told you that the electrons are emitted from the electron gun, and then you have sample, and in between you have the optics. With what it will do, that most of the times the main aim is to deflect the trajectories of the electron beam. So let me try to make it a bit clear with the help of this simple explanation. So what will happen if you have the important component? What do you need to have in the case of the electron microscope? You need to have one electron gun. So that is the first thing you need to have, because if we don't have electrons, then how we can scan a sample with the help of the electrons? That is the first parameter. And the second, if you don't recall all the components, then I will say that okay, these are lenses. No, these are lenses are to direct the electrons from one place to the other place. No? And then the third thing, you need to have the sample, and then after that you need to have this, the detector. No? So these are the important things which you need to have. So how does it work? You have electrons which are coming from somewhere. These are the electrons. And then what will happen? These electrons will hit onto the surface of the sample. This is sample. Now what will happen? That the electrons, they are scattered in some direction, and we need to have a detector for that. Detector D. So overall, what we are trying to do here in this case, that electron, how to produce the electrons, that is as simple, because the electrons are produced by with the help of the thermionic emission from the tungsten filament. So what you would have a tungsten filament which can emit the electrons. And once the electrons are emitted, now our next task is to direct the trajectory, because in normal cases the electrons will be going in all the directions. How to define their trajectories? These trajectories are defined with the help of the lenses. And we have also to understand that what kind of lenses are these. So if I pull up this thing, so it is written first condenser lens, spray aperture, second condenser lens, deflection coil, the final lens aperture. Then you have the all this stuff over there. So these lenses, if we would like to make it clear that these are different from the optical lenses. So normally what will happen, if you look at your hand, sometimes there are some people, those who tell future, the palmists, so that they have a magnifying glass, okay, and this is a kind of lens, and this lens is basically an optical lens. In this case, you don't have the optical lens, these will be the electromagnetic lens. So we have to make it clear that these are not the optical lenses, these are the electromagnetic lens. So what will happen in case of the electromagnetic lens, that you apply the electric and magnetic field which can deflect the trajectories of the incident electrons according to the principle that how much force will be exerted by the external electric or magnetic field on the charge carrier. So that can be given with the help of the Lorentz law. So then you have the sample, and then you have the detector. So this the detectors. So what will happen in case of the detectors, that you would have the emitted electrons. Now one concern remains, that what will happen at this point when the electrons, they interact with the sample, what type of the processes will be there. So this will be answered with our next slide in this case. So what we are trying to do, we are just coming across this point, which is we would like to highlight this point in this case, that what is happening in this case, in the case of the sample, when the electrons they are interacting with the sample. So let's come to that point in this case. So let me pull up the next slide. So these are the events. What will happen here? Okay. So the electron beam which is incident, then there will be the secondary electron emission. There may be also OJ electron emission. There is also a possibility of backscattered electron, and then there is also a possibility that the incident electron produce the characteristic X-rays, or then you have these continuum X-rays, and then there is also a possibility of cathodoluminescence. And there is no transmission in the case of the scanning electron microscopy. Only these electrons which are either scattered backwards or at certain angles with respect to the sample surface, they will be detected in this case. So if you recall that, let's take one by one. What are the secondary electrons and what are the OJ electrons? We have different topics which are dedicated to these electrons, and let me pull up the first thing, that here you would have the electrons, they interact with the sample. How they interact? So one, there is the possibility of the backscattered electrons, the electrons which are scattered in the same direction. These are due to the elastic scattering. In case of the elastic scattering, you know that momentum and the energy, kinetic energy of the incident particle, both are conserved. So what will happen, that in that case you would have the backscatter, but you need to have a different detector in each case. And in majority of the cases, the secondary electron emission, the secondary electrons which are emitted in this case, these secondary electrons are responsible for producing the images. Okay, these electrons, secondary electrons, are responsible for producing the images in case of the scanning electron microscope studies. So what will happen, that how these secondary electrons are emitted? Let me show you that. Okay, what is inside the atom? You have the nucleus which contains protons and neutrons, and then the electrons, they are revolving around the nucleus. So there are definite shells like K shell. Everybody knows that at M.Sc level. So you would have K shell, L shell, and then here you would have 2 electrons, and then here you would have 8 electrons. Okay, 8 electrons. So what will happen, that incident electron can knock out one electron. For example, if the incident electron is coming like this, then it may knock out this electron, and this will be the secondary electron. This will be the primary electron. The itself, the fundamental electron is the primary electrons, and then these are the secondary electrons which are emitted from the cells of these structures. So for example, if you have when there is a vacancy created in this cell, this vacancy, because the incident electron, they hit the K shell electron, so this electron will be knocked out, producing the secondary electron. Then this vacancy can be filled by the higher cell electron. So what will happen? So you would have higher cell electron, and then you would have emission of the characteristic X-ray in this case. And sometimes these radiation, they won't come, but they will knock out the outermost electron in this case. And these electrons, they are known as the OJ electrons, because they are triggered by the characteristic X-rays, which of course they transfer the characteristic X-rays, they transfer their energy to the electrons in this case. So that we have discussed in this case. And then you have the brimstone, that okay, the electrons, when they are deaccelerated, then you would have emission of the secondary electrons. Cathodoluminescence means that okay, what will happen in that case? You can see that if the sample is having this much penetration, then you have the cathodoluminescence, means that there will be some visible radiation which will be emitted during this process. So if you look, these are the systematically, we have defined all the patterns of these the secondary electron emission, how they are produced, or characteristic X-ray. Systematically, these are the backscattered secondary electrons. These are the OJ electrons, and here you have X-rays which are emitted during this process, and this all the things which we have during this process. So now when this process is completed, these type of images are produced with the help of the scanning electron microscope. But the second image which is there, so these type of the images, for example, you can have a scale which will tell us the resolution of the particular sample, that how you can resolve that sample in this case. So then you would have these type of the images which are produced in this case, and then the final one is, these are the references which we have used in this case. And that's a question, that can we analyze this image after looking at the lecture and the details given in the lecture? Are the students able to solve this problem? Look at this image and look at different points of this image. So what I would suggest, the first step would be that you should look into the problem. So for example, if you are look at the scale. First thing is that look at the scale, and once you are familiar about the scale, you know 50 micrometer, then you can know that what are the different components and what would be the resolution here. Okay, you can, if you want to measure this distance from here to here, if you want to measure this one, then you can do that. Of course, that can be done with the help of this scale. Okay, you put that equivalent scale, and then you will be able. There are different softwares also which can be used to analyze the images. These are called the JS softwares. But right now we are not concerned at master level. We are just going to give you a small glimpse as how does it work. That we can direct. These are the references, and then you have this advanced study references. You can have this information in this case, so you can have all the details in this case. But what we have covered in this case, if you recall, then we have just discussed that how does it work, introduction to the electron microscopy, difference between the optical and electron microscope, experimental configuration of SEM instrument, and references. So with the more study we can come to the next chapter. Thank you very much for listening and looking at this video. I hope that it will be helpful.